Now we're going to talk about process of generating glucose from pyruvate. So the opposite of what we've been talking about with glycolysis, we're going to talk about a process called gluconeogenesis, which takes two pyruvates and it converts it back to a glucose. Now, um, most of the enzymes that we use in this process of gluconeogenesis are the exact same enzymes we used in glycolysis. And the reason why we can use those exact same enzymes is because the energy difference between the reactants and products for much of glycolysis, there isn't much difference, right? So we remember that enzymes just form the equilibrium more quickly. So if we feed pyruvate into this system, it's going to drive the equilibrium backwards so that we can generate reactants instead of products. But that's not the case for three main areas in the 10 reactions of glycolysis. And you may have noticed that these three reactions are our kinase reactions: pyruvate kinase, phosphofructokinase, and hexokinase. These reactions have such a huge energy difference between the reactants and products. The equilibrium is shifted so much towards products that we can't just use these same enzymes to generate much of the reactants if we want to drive this reaction backwards. And I kind of think of the energy diagram of glycolysis as almost like a river, right? This river is going downstream and we're trying to canoe along it. Now, if we're trying to canoe this river backwards through gluconeogenesis, we can do that. We can paddle upstream for a lot of this river, but there are three locations where really we have waterfalls that we can't paddle back upstream. We're going to have to spend some energy and go around these waterfalls to get to the top of them so that we can continue paddling upstream. And these waterfalls are really just big energy differences between reactants and products. So let's look at how these waterfalls are going to work. If we look at the 10 reactions of glycolysis, we can focus in on the three waterfall locations, the three kinase reactions, and we can see how we're going to try to get upstream. We're going to have to spend some energy to go up these waterfalls. Now the first waterfall is the most complicated. We're trying to go backwards where pyruvate kinase went forwards. And we're going to use two separate reactions in order to convert pyruvates back to PEPs. The first reaction is catalyzed by pyruvate carboxylase, which uses ATP and carbon dioxide to generate a molecule of oxaloacetate. We'll see oxaloacetate in the Krebs cycle. It's really just pyruvate with carbon dioxide attached to it. Now, oxaloacetate is then acted upon by PEP carboxykinase and the action of GTP to form our PEPs, which lose a carbon dioxide, going back to the 3-carbon PEP. Now, what's the cost of converting two pyruvates back to two PEPs? Well, you might think the cost is 4 ATPs, but I'm going to try to convince you it's only 2 ATPs. So let's convert two pyruvates to two oxaloacetates. That's going to use 2 ATPs. 2 oxaloacetates back to 2 PEPs. That's going to use 2 GTPs. So the total is 4 right now. However, I would argue that only 2 of these ATPs is actually wasted. The other 2 high-energy phosphates are bound into our carbon chain and are present in our PEP. And so those aren't wasted. And one way to test whether high-energy phosphates are wasted or not is to try to just go around in a little circle here, backwards and forwards and backwards and forwards. If high-energy phosphates are wasted, then you'll see them being wasted. If not, then you'll regain them when you go back the other way. So 2 pyruvates to 2 PEPs costs 4 high-energy phosphate bonds. But then if I go 2 PEPs down to 2 pyruvates, I can get 2 of them back as ATPs. Lose 4, gain 2. Lose 4, gain 2. So I'm really only wasting 2 ATPs here. Now let's go backwards. We can use the same enzymes as we turn our 2 PEPs to 2 of these, to 2 of those, to 2 of those, to 2 of those, all the way to fructose 1,6-bisphosphate. Now there's a couple places where ATP comes into play, but these aren't wasted ATPs, right? As we go from 2, 3 phosphoglycerate molecules to 2 1,3-BPGs, I use 2 ATPs. But I could get them back just by converting it backwards. If I go back and forth here, there's no waste of ATP, no waste of NADH. But let's look here. Here's our second waterfall where we're trying to convert fructose 1,6-bisphosphate to fructose 6-phosphate. We can't use phosphofructokinase. We're going to use fructose bisphosphatase, which uses one of the high energy phosphate bonds to move us back up the waterfall to the top. And this is a wasted high energy phosphate bond, because if we go backwards and forwards and backwards and forwards here, we are losing an ATP every time we go through that reaction. Okay? And that ATP is lost as just an inorganic phosphate. We also have a loss of 1 ATP when we go from glucose 6-phosphate back to glucose. Again, lost as an inorganic phosphate by the action of glucose 6-phosphatase. Now, that's it. You know, these enzymes are using that one high energy phosphate bond to drive this reaction up the waterfall at these two places. So to make one glucose, it's going to cost us an ATP here. It's going to cost us an ATP here. And it's going to cost us 2 ATPs here. So our total waste of ATP as we convert 2 pyruvates to 1 glucose is 4. So why do this? What's the point of it? You know, if it's going to cost 4 ATPs, that seems like a lot, because burning 1 glucose only generates 2 ATPs. Why in the world would we ever drive this glycolysis backwards in gluconeogenesis? Well, usually this occurs in starvation or in times of plenty. It turns out proteins can be broken down into pyruvates. And so if you eat a lot of protein and you've got a lot of protein floating around, your body senses that and it suggests, well, maybe we should store some of that energy. And so it will convert the protein energy present in pyruvates back to glucoses to store it as glycogen for times when it's going to need that. And it's willing, your body is willing to waste some ATPs in order to store that energy for the future. Now, that's in times of plenty. Now, in starvation, times of need, it turns out that your body undergoes gluconeogenesis also. And that's because your brain is hungry for glucose. In order to keep your brain going, you've got to supply it with glucose. And so your body will sort of break down the proteins in your body to generate pyruvates, to feed them into gluconeogenesis, to generate free glucose, to feed your brain to keep your brain going, because your brain's the most important thing. And so in starvation, the rest of your body breaks down and wastes away to generate glucose to keep your brain going, and it is a waste of energy in order for that to happen, but it's important to keep your brain running. Okay, now how is this process between glycolysis and gluconeogenesis regulated? Well, not surprisingly, it's regulated at the rate-limiting step that is catalyzed by phosphofructokinase. And it turns out that this regulation is influenced by the action of hormones. The two hormones that really influence whether we're going to do glycolysis or gluconeogenesis are insulin and glucagon. So let's imagine that you eat a candy bar and therefore have a lot of sugar in your bloodstream. That promotes the secretion of insulin. Insulin is a hormone that activates this enzyme called phosphofructokinase 2. Phosphofructokinase 2 converts fructose 6-phosphate into this regulatory molecule, fructose 2,6-bisphosphate, sort of the cousin of the fructose 1,6-bisphosphate that's present in glycolysis and gluconeogenesis. Now, fructose 2,6-bisphosphate is a regulatory molecule. It will activate phosphofructokinase and inhibit fructose bisphosphatase. So it's activating the glycolysis enzyme and inhibiting the gluconeogenesis enzyme. So the net result is glycolysis occurs. So let's recap that. You eat glucose, produce insulin. Insulin activates PFK2, which produces fructose 2,6-bisphosphate, which activates glycolysis and inhibits gluconeogenesis. So all that glucose that you ate is going to be broken down. Yeah. Now, what if glucagon? What if you have low levels of glucose and you need to make some for your brain? Glucagon is going to be generated to generate it for your brain or to generate glycogen. Now, glucagon inhibits phosphofructokinase 2, which decreases the levels of this regulatory molecule, which prevents the activation of phosphofructokinase and prevents the deactivation of fructose bisphosphatase. So when glucagon is present, you have low levels of the regulatory molecule, which promotes gluconeogenesis and prevents glycolysis from occurring. And that's how we regulate whether we're doing glycolysis or gluconeogenesis at this rate-limiting step by the regulation of this regulatory molecule, fructose 2,6-bisphosphate.